Hi everyone, my name is Wenming Zhang. I'm a research scientist in Professor Sherry Kram's and Professor Oliver Martinez's lab in transplant immunology. Today, I will be talking about the project that I've been working on during the past year in our lab, which is high-resolution natural killer cell phenotyping by mass cytometry in EBV positive and EBV negative individuals. Epstein-Barr virus, EBV, is a broadly disseminated virus that infects over 90% of the world's population. EBV-associated complications are particularly common in pediatric transplant recipients. In many studies, EBV viremia exceeds 30% of subjects. Natural killer cells are lymphocytes of the innate immune system that respond to virally infected cells via cytotoxic pathways or production of cytokines. It has been reported that NK cells play an important role in the control of EBV infection. Previous work in our lab has shown that NKG2A positive NK cells are enriched in the NK cell population that can recognize and respond to B cells that are latently infected with EBV. However, the interrelationship of latent EBV infection and in vivo NK cell populations in transplant recipients was yet to be investigated. So the purpose of my study was to first determine whether EBV alters the proportion of NKG2A positive NK cells in vivo, and secondly, achieve high-dimensional phenotyping of the NKG2A population associated with EBV infection. For this study, we used the CTOTC06 cohort, which is a prospective, multi-center, observational trial of pediatric transplant patients. The workflow is shown in Figure 1. Peripheral blood NK cells in 3 to 5 million PBMCs were obtained 6 to 9 months post-transplant from 32 participants. And the cells were then stained and analyzed on CyTOF with a 48-marker mass cytometry panel we established in the lab that include multiple activating receptor, inhibitory receptors, as well as other functional markers of NK cells. And the panel is as shown in Figure 2. The data was normalized with Fluidigm software and gating of the CD56 positive and KG2A positive NK cells was completed in Cell Engine. With a Wilcoxon rank sum test, we observed a significant increase in the proportion of NKG2A positive NK cells in EBV positive individuals. A further analysis with flow sum and consensus cluster plus clustering in R helped us identify 10 unique cell populations of CD56 positive NKG2A positive NK cells, including subsets that are characterized by differential expression of markers including CD56, CD16, CD2, NKG2A, NKP46, NKP30, 2B4, Siglet7, perforin, and granzyme B. Understanding the NK cell subsets that control EBV infection will lead to improved therapies for transplant recipients. That concludes my presentation. If you have any suggestions, comments, or questions, please feel free to email me at wenmingz@stanford.edu. Thank you.